Good morning, I am Madhurima Datta, a Zoology and Biology educational content creator. Now's content for B.Sc and M.Sc in Zoology Honours, Microbiology Honours, Physiology Honours, Biotechnology Honours, Molecular Biology Honours, and also in Life Science. And exams for CUT PG exam, GATE exam, DBT exam, IIT JAM exam, and CSI NET Life Sciences exams. Paper Developmental Biology, Unit 1, Introduction of Developmental Biology, and today I will discuss the introduction of the Developmental Biology. Let's go. Introduction to Developmental Biology. At first, we will know about the embryo. What is the embryo? Every multicellular organism has to function even as it builds itself. It develops in the time between fertilization and birth. The organism is known as embryo. The concept of an embryo is a staggering one. Means embryo is an unborn or unhatched animal in its earliest stage of the life, and this phase spans the entire duration between fertilization and birth or hatching. Next point, multicellular organisms do not spring forth fully formed, rather they arise by a relatively slow process of progressive change that we call development. In most cases, the development of multicellular organisms begin with a single cell and egg cells that has completed the process of fertilization, that is referred to as a zygote. Meanings, the gradual progressive process through which a simple cell that is grows and transforms into a complex and fully formed multicellular creature is called developmental process. And what is the zygote? Zygote means the starting point of life. A single fertilized egg cells is called zygote. Next, embryology. What is the embryology? The study of animal development has traditionally been called embryology after that phase of an organism that exists between fertilization and birth. Means the scientific branch of biology that focuses on studying animal during their embryo phases is called embryology. Next, an animal life cycles. Most animals, whether earthworm or eagle, termite or beagle, pass through similar stages of development. That is, fertilization first occurs, then cleavage, then gastrulation, then organogenesis, then hatching or birth, then metamorphosis and gametogenesis. The stages of development between fertilization and hatching are collectively called embryogenesis. First step is fertilization. Fertilization involves the fusion of the mature sex cells, the sperm and eggs, which are collectively called the gametes. And the fusion of the gamete cells stimulates the egg to begin development and initiates a new individuals. And then the subsequent fusion of the gamete nuclei, that is the male and female pronuclei, each of which has only half the normal number of chromosome characteristic for the species, gives the embryo its genome and the collection of genes that helps instruct the embryo to develop in a manner very similar to that of its person, of parents. Means in short form of the fertilization is the sperm and egg cell fuse together first, then the fusion activate the eggs development. Means the sperm and egg cell fusion activate the eggs development, then combined DNA creates the complete genome blueprint. Next process is cleavage. Cleavage is a series of mitotic division that immediately follow fertilization. During cleavage, the enormous volume of zygote cytoplasm is divided into numerous smaller cells called blastomere. By the end of cleavage, the blastomeres have usually formed a sphere known as blastula. Means of the cleavage, that is, the single zygote divide rapidly at first, then the resulting smaller cells are called blastomere, and the cells eventually form a hollow sphere shape called blastula. Next process is gastrulation. After the rate of mitotic division slow down, the blastomere undergo dramatic movements and change their positions relative to one another, and this series of extensive cell arrangement is called gastrulation, and the embryo is said to be in the gastrula stage. As a result of gastrulation, the embryo contains three germ layers. First is endoderm, ectoderm, and mesoderm that will interact to the generate the organs of the body. In short form of the gastrulation is that is the cells slow down division and move around. Next three distinct cellular layer are formed, that is the ectoderm, mesoderm and endoderm. Ectoderm is the outermost layer, mesoderm is the middle layer and endoderm is the innermost layer. And these three layers construct the body organs. Next, organogenesis. Once the germ cells are established and the cells interact with one another and rearrangement themselves to produce tissue and organ. This process is called organogenesis. Description of the organogenesis. Chemical signals exchange between the cells of the germ layers resulting in the formation of specific organs at specific sites. Certain cells will undergo long migration from their place of origin to their final location. These migrating cells include the precursors of blood cells, lymph cells, pigmented cells and gamete cells. Means that the process where cells interact, move and rearrangement themselves to create tissue and organs inside a developing embryo. This process is called organogenesis. And how the organogenesis works. When cells send chemical signals to each other, so they know exactly where to go and what organ to form. Some cells travel long distance from where they started to reach their final destination. Next stages is larva. In most species, the organism that hatches from the egg or is born into the world is not sexually mature. The organism needs to undergo metamorphosis to become a sexually mature adult, and the organism needs to undergo metamorphosis to become a sexually mature adult. In most animals, the young organism called larva, and it may look significantly different from the adult. Next, somatic cells and germ cells. These cells are the precursor of the gametes. The gametes and their precursor cells are collectively called germ cells, and they are set aside for reproductive function. All other cells of the body are called somatic cells. And this separation of the somatic cells and germ cells is often one of the first differentiation to occur during the animal development. This is the very most important part for the MCQ of GATE. Next, gametogenesis. What is the gametogenesis? The biological process by which diploid or haploid precursor cells undergo cell division and differentiation to form mature haploid gamete is called the gametogenesis. A sexually mature adult frogs, and they have a male frog and female frog. They produces male gametes and female gametes, that is called sperm and oocytes, then fertilize, then produce zygote, and then produce morula, then blastula, then blastocele, then blastopore. And after blastopore, they are in their zygote, their organogenesis occurs and form ectoderm, mesoderm, endoderm. And after this process, hatchings occur. Then first is immature larval stage, and then that is second immature larval stage where gonad is formed, and then sexes are separated and sexually mature adults are formed. Here, what is the blastocele? The fluid-filled central cavity located inside the blastula is called blastocyst. And what is the blastopore or function of the blastopore? The external opening of the primitive gut that forms during the gastrula stage of the embryonic development is called blastopore. Now the topic has ended. For all video of B.Sc and M.Sc in Life Science, Physiology Honours, Biology Honours, Zoology Honours, Biotechnology Honours and Microbiology Honours, please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Datta. Thank you everyone.